and welcome for this video. So in this video we will talk about diagnosis of jaundice and using the tear fluids. We already aware of that we secrete so many body fluids in form of sweat, and that sweat's having some metabolic byproducts, and basically those are the waste itself, major portions of that. But on the same time we secrete the tears also, and these tears having various antibiotics and various like special biomolecules also in some of the cases. But majorly in the tears those are lysozymes, IgGs, and some kind of minerals also. Yeah, so minerals. So in that way we secrete. And why? Because antibiotics like IgGs and other things like in lysozymes, just because of any, so they can inhibit any kind of bacterial growth over these eye, so it can protect our eye. But in some of the cases, like patient is suffering from any particular kind of any kind of disease, so then metabolic products of that diseased persons may have some various, so they secrete some specific biomolecule also. So if we able to extract, or if we able to detect such kind of biomolecule from the tears, then we can diagnose a person with that particular disease. Now that has been done in one of the research, where what they have done, so they formulate silicon dioxide nanoparticles, and then this silicon dioxide nanoparticles having a thin layer of gold over these silicon dioxide nanospheres, and then the nanospheres having these gold thin layers bombarded or coated with the specific gold nanoparticles. And those gold nanoparticles, so it is called the SEARCH technique, surface enhanced Raman scattering. So this video I can't talk about the Raman because it's take then too long time to talk about. But the Ramans are the specific signals which is emitted by each and every kind of biomolecules, and these Raman signal are specific for each and every kind of chemical moieties, like each and every kind of chemical molecules, atoms, even if it is a biomolecule, like any kind of organic compound, they emit specific kind of Raman signals. So in that way, basically those are identity. For example, Aadhaar card is identity in India, and in US there is a one different ID for each and every person. At the same time, each and every molecule or atoms, they have some specific identity, and that identity is driven by the specific signals which is emitting by them. So Raman is one of them, and Raman is very highly specific signal. So what happening, so when they coat the silver dioxide with these gold nanoparticles, and why? Because it enhances the Raman scattering. So whatever the signal is coming from this biomolecule get enhanced, and once they enhance, it can be detected easily. So they coat on a glass slides, and then they cover that glass slide with another glass slide, and they make one input so they can absorb the tear or tear samples. Once the tear flow through those those nanospheres, silicon dioxide nanospheres, so any patient with the jaundice, like anyone who has a jaundice, they must have bilirubin secretion in a higher amount. So those bilirubin also secreted in the tears from that patients. So these bilirubins are attached with those gold nanoparticles, and once they attach with the gold nanoparticles, they start emitting, like after the laser bombardment or after scatterings or after scanning, they emit some specific Raman signals, and those Raman signal is enhanced by gold nanoparticle itself, and it can be detected by any diagnostic equipments. So once it detected, then we can confirm that the patient is suffering from jaundice or not. So it is more like a qualitative diagnosis, like we can discriminate between the jaundice disease person and non-jaundice person, but we cannot say like extent of this jaundice in that particular patient for till now. But definitely maybe some other optimization process we need to do with that nanoparticles or such things, so maybe that thing is also doable. But for now we can detect like whether the patient has been suffering from the jaundice or not, not by this. So bilirubins are one of them. Another biomolecule also in the is a biliverdin. So bilirubin and biliverdin, these are the two things, but especially in the jaundice case, the bilirubin concentration become higher, and it can start secreting from all over this this sweat or secretory biomolecule, or secretory, we can say the secretory fluid from the body. So tears is one of the secretory fluid. So from there also, even in the some cases, I'm not sure if they we maybe talk about in some different way. I'm just talking about even the breastfeeding also. In breastfeeding time, if somehow if anyone get jaundice, so then there is a high chances also. So yeah, so these are the things which have been discovered. So it's one of the great discovery during this diagnosis process. And apart from that, what is the search is very specific and very enhanced. So the diagnosis is very near to the accuracy, or more accuracy, rather than other IVD devices. IVD devices are the in vitro diagnosis devices. So yeah, so even these also, this setup can be done in the IVD form also, and these people are doing it, but this is little bit more specific, just because of the surge signals. In IVD there is no surge signal, there is only the gold nanoparticle coated with the antibiotic, and they attach with these bilirubins, and then ligand and antibody interactions, and then they can detect that. But in the case of the surge, even the smallest fractions of bilirubin can be detected with the surge based diagnosis systems. So that is one of the great thing in that. So for this video, that's it for now, and in the next video we will meet with some another topic. Till then, bye bye, take care.